Thank you, Chairman, for the introduction, and I also want to thank the organizing committees for putting on this wonderful conference for us to all learn about African swine fever virus and the strategies to deal with it. Today, in my presentation, I would like to show you the dietary role in managing this African swine fever virus threats. To fight your enemy, you have to first understand your enemy. To live with this African swine fever virus, we have to first understand this virus. I believe there are several really unique features about this virus that if we learn, we can really, using that, to develop strategies against this virus. The first very unique features about African swine fever virus is the target cells. We know this African swine fever virus only infects the monocyte cells of pigs, which are the macrophage cells and the dendritic cells. Those are immune cells which sits inside the pig's lymph node or underlayers of epithelial layer. On the other hand, the epithelial cells are not the host cells, meaning the African swine fever virus, they will not infect those epithelial cells. This is very interesting and important because if this is the case, and if we can maintain the epithelial layer healthy and not leaking and no damage, we can potentially prevent African swine fever virus to penetrate through these epithelial layers and to get in contact with its target cells, which are again the macrophage cells and the dendritic cells, which will help us to really reduce the infection or the chance of infection. The other unique features about this virus is African swine fever virus. The pig doesn't seem to produce neutralizing antibodies against this virus, which is one of the reasons why this virus are so deadly. However, there are some reports showing if you inject the serum from a recovered pig to an infected pig, it will help to increase the survivability of that pig. This is interesting as well because this is showing there is something in the serum that can really help with the immunity of a pig to deal with this African swine fever virus. The third point of this virus is it really needs the pig to develop a so-called cytotoxic CD8 T cell cellular response, so for it to fight against this virus to reduce transmission and increase survivability. This picture over here really illustrate the first point that I made from on earlier slides, which are the importance of epithelial barrier health and its role in reducing African swine fever virus infections. As you can see on the left side, when we have very healthy epithelial layers, we can potentially prevent the African swine fever virus penetrate through the epithelial layers and get into contact with its target cell, which are macrophage cells and the dendritic cells, showing here in blue and the purple. And potentially that will help us to reduce infections, or at minimum to increase the minimum infectious dose are needed by this virus to infect the pig. On the other hand, if we have some damage on this epithelial layer, whether that's a gastric intestinal damage, diarrhea-related damage, or a damage related to another virus challenge caused a lung lesion, for example PCV2 or APP, nevertheless, this will really increase the African swine fever virus penetration into the donor layer of the epithelial layer and have them to get in contact with its target cells. And this really increases the chance of African swine fever virus infections even by a low dose exposure. So the importance here is we really need to focus on the epithelial barrier health to reduce the African swine fever virus infections. There are some report or some paper published in the literature really support this idea. This is a paper published by Dr. Sandra Bloom's lab, who is a leading scientist in Germany on African swine fever virus. This is a paper published by her lab where they looked at the minimum infectious dose of African swine fever virus to unhealthy animals and immune compromised runt animals, where she concluded when she oral nasally administered 100 units of this virus, she could not infect a healthy pig. However, if she used even lower dose, 10 units, she was able to infect an immune-compromised runt pigs. And also, she mentioned in this paper that the first pig getting infected are having some gastrointestinal issues, which she think might contribute to this low dose infection of pigs. So from what we learned, or also from what we had from this paper, we can see the herd health and this mucosa integrity, especially the epithelial layer, its health, its integrity, and also the proper immune response are very keys to control this African swine fever virus. Okay, now you may want to ask, okay, what are the things that can help us to increase the herd health and preventing for example, the epithelial layer damage? There are several factors can benefit the pig health. This is a paper published in 2006 by Dr. Duane, who is a Canadian vet, where he looked at a large data from an epidemiological study looking at all the nursery pigs in Manitoba, and by comparing all those nursery barns, what are the factors that they're doing differently that really can impact the herd health. Then he concluded the farm biosecurity, disease status, farm management, and also nutrition, all are the factors can help to benefit the herd health and also pigs' survival. And here, I pointed out here, the nutrition, especially inclusion of plasma in the nursery diet, really significantly improved the pig health and the survival. On the right side on the graph, you can see the odds ratio of including plasma for increasing the pig's health and survivability by including plasma in the first diet and the second diet. Odds ratio for them are 13.5 and 4.9 respectively. They are all even higher than the reference, for example, higher weaning weight, the odds ratio for higher weaning weight. We all know higher weaning weight will increase pig survival or pig health, but this data from Dr. Wayne really shows the importance of nutrition. It's probably more important actually than the higher weaning weight from this piece of the information. But nevertheless, this is really showing you there are several factors can improve or can help with pig health, which we all have to really focus on for your pigs if you are in the African swine fever threat zones. And also nutrition are very, very important as well. When we talk to an epidemiologist or a virologist, talk about the key strategy to limit the virus infection, we often really are, spending our time try to focus on minimum the exposure and also reduce the transmission, the possibility of transmissions, which are very true, and you should probably spend 120% of your time try to do that, really minimize the exposure and transmission, which are including factors like biosecurity, increase your biosecurity, try to have a better management of your farm or your employees, pig flow, testing and monitoring. Those are things you need to do, and you probably need to do 100% or 120% of your effort. However, we do believe there are another side of this equation, which are pig itself. the house of a pig or herd health, or the epithelial layer health, those are probably important as well, as I just illustrated from previous slides. So we do believe if we can improve the herd health or the gut health of the herd, we can also help the pigs to deal with this virus. I'm a nutritionist, so I want to, what I often ask myself is there any dietary interventions or strategy that we can use to optimize the herd health and the immunities to help the pigs to fight against this virus? The answer is really yes, clearly, because we have a lot of data to show nutrition can impact gut health and also impact the immune system. In this way, the diet definitely can help the pigs to deal with this virus. What are some of the examples of dietary ingredient or diet regulating immunities? Here I'm giving a few examples here. Those are functional proteins, which are essentially and part of the immune system, right? And like the IgGs or interferon, those bioactive proteins, they're essentially a part of the immune system. They sure can regulate the immune system. For example, the IgGs, it can neutralize the pathogens. Also, although as I mentioned, the African swine fever virus, the IgGs, the antibodies, will not neutralize this virus, so it doesn't work that way. However, the immunoglobulins do facilitate more efficient immune response. In that way, it can help. The other proteins like interferon gammas, we know it itself has antiviral properties. There are data suggesting it actually can inhibit African swine fever virus replication in the host cells, and also there are data suggesting it can prevent the penetration of the virus into a target cells. Also, it facilitates some specific CD4, CD8 T cell response, which are very important for controlling this virus or helping pigs to survive from this virus. Also, there are other proteins can really help with the gut health as well. So again, this is really illustrating the dietary component or ingredient can do impact the immunity of a pig. Certainly, as a company, we produce plasma proteins, and we in the past have done a lot of plasma challenge trials with plasma. As you can see here, we have run the trials with pigs, calves, and poultry, and also aquaculture species on all kinds of challenges: E. coli challenge, Salmonella challenge, or virus challenge like PRRS challenge or PCV2 challenge. Under those challenge study, we always see the pigs or other species always have a better diarrhea score, better average daily gain, better survival, and feed efficiency when they are fed with plasma functional proteins. We believe the reason why we see that result is because the functional proteins in plasma as an essential part of the immune system really can improve the efficiency of an animal to deal with the disease challenge, and in that way it increases the health of an animal and improve the performance. So coming back to this slide again, and this is really showing you how we believe plasma protein really helps to reduce the risk of virus infection. It's really through a better gut health. We have a lot of data on plasma protein. Its role in the epithelial layer health. We have a lot of paper in the literature showing plasma protein can improve tight junction, improve barrier function, reduce inflammations, and actually can repair the damaged epithelial layers as well. So the plasma protein do play a very unique role in keeping the epithelial layer healthy, or even when the damage happens, the plasma can actually help to restore those damaged epithelial layer back to normal quicker than control. So this is a very unique role plasma can play to improve gut health. So in this case, in African swine fever virus threat, we believe plasma protein really can help to at least reduce minimum infectious dose, or actually increase the minimum infectious dose needed for African swine fever virus to infect the herd. Or to reduce the transmission rate between pigs to pigs because they have a better gut health. You might need a higher dose for pig to get infected, so it can actually reduce the transmission rate between pig to pig. With that in mind, we actually run a trial at the federal facility, CRASA, at Spain. It's a bio-level 3 facility where they are specialized in virus research, especially African swine fever virus. This is the result of this particular research. The goal of this research is really looking at the role of spray-dried porcine plasma on pigs contacted with pigs we artificially challenged with African swine fever virus, and to see how by feeding strategies impact those artificially infected pigs and also the contact pig. Do they behave, does the diet actually increase or reduces the transmission rate between pigs in the same pen? And those are the goals of this particular research. This is the timeline for this research. As I mentioned before, we have 2 dietary groups here. Group A contains 8% spray-dried porcine plasma. Group B is a control feed. We had 24 pigs in this research. 12 pigs were allotted into Group A in the room, and another 12 pig was allotted to Group B to another separate room. Again, this is the timeline. All pigs were brought into this facility 4 days before the trial starts, and to give them time to accommodate to this facility. On day 0, we select 2 pigs from each group, randomly selected them, and then we inject the live virus, the African swine fever virus, to these 2 pigs. We call them Trojan pigs. And then we observe those 2 pigs for 2 days. After confirming they all got infected with African swine fever virus, we put them back into their original pens. Then we studied those 2 pigs and also the contact pigs by measuring their blood, nostril swab, rectal swab to see whether those pigs get infected or not. The contact pig get infected or not. Then on day 6, what we find is those 2 Trojan pigs from, actually 4 Trojan pigs from both groups all developed high fever. Under the lab protocol, once they reached 42 degrees, long and heavy fever, we have to euthanize them because of the lab procedure. So we euthanized all 4 pigs from the 2 groups on day 6. Then we followed all the contacted pigs in those particular groups all the way to day 18 to see how many pigs get infected and how fast they get infected. To our surprise, very surprising, none of the pigs from either group ever had developed African swine fever infections. So, because of that, we decided to extend this research a little longer by introducing additional Trojans, additional infected pigs. So in this case, on day 19, we brought in another 3 Trojan pigs. So we randomly selected 3 pigs among those remaining pigs in those 2 groups from each group 3 pigs. We randomly selected them, and then again we injected them with African swine fever virus with about 1,000 doses of those Georgian strain, and then we put them back into the pen immediately. And then in this case, we did not euthanize those pigs even though they developed high fever. So we left those pigs in the pen all the way until they naturally died. The idea is to increase the threat of the threat of the virus and increase the infections. And then we can learn does the diet impact the rate of transmission among the pigs. Again, for the second group, we followed them all the way to day 31. At day 31, that's when we have to stop the study, and we euthanize all the pigs, and we collect the tissue sample, blood, nostril samples, and rectal samples for analysis. This slide shows the temperature of the group A, of those pigs fed with 8% spray-dried porcine plasma. As you can see on the bottom, you can see those are the date of the timeline of the study, and then each line on the top tracks the temperature of particular pigs. As you can see on this top side, after we inject those 2 children pigs with African swine fever virus, they quickly develop high fever, and by day 6, we had to euthanize those 2 pigs. Then on the right side, you can see we have another second round of Trojans, which are those 3 pigs we injected with live virus. As you can see, they also developed some fever, some high fever as well, which are shown in the red. One pig reached a really high fever early on, and the other 2 pigs reached a high fever, then seems to stay stabilized. The other color lines over here are those contact pigs. As you can see, those contact pigs, they didn't really develop any high fever until up to the end of the study. If we compare to the control group, as you can see here, the control group, again, similarly to the plasma group, all the challenged pig, they all developed high fever pretty quickly after injection. However, the contact pigs in the control group, those pigs seems to develop the high fever much quicker in the early days compared to the control group. This slide shows the average temperature of the pigs, of those contact pigs in those 2 dietary groups, by comparing those two groups' temperature. As you can see on the slides, the blue line represents the plasma group, and the yellow line represents the control group. As you can see, for most of the time, and the average temperature of the 2 groups are below 40 degrees, so they don't have any fever. However, at the end of the study, the control group does develop a high fever more quicker than the plasma group, which are suggesting that the control group are being infected quicker and developing the clinical sign much quicker compared to the plasma group. Now, we also, collected all the samples from blood and nasal and rectal swab, and also, tissue samples, and and then later on we analyzed those samples with PCR test and try to look at the DNAs or the genetic materials in those samples to see to see the viral load in those samples. And this slide really shows the children pigs 6 days post injection, where what is the viral load between groups. As you can see on the top is the plasma group, on the bottom is the control group. And again, this is a PCR test where we tested the CT value of a specific sample, blood sample, nasal swab, or rectal swab. As we all know, the CT level, the lower the number is, the higher the DNA copy is. I actually made a conversion from the CT value to the DNA copies. As you can see, on the plasma group versus the control group, either in the blood or nasal swab or rectal swab, the plasma group pigs, even with this injected children pigs, the plasma group pig really had a lower DNA copies in the blood and nasal swab and rectal swab, and the fold differences between control group are quite significant, 44 times, almost 60 times or 8 times in the rectal swab. So, again, even in these children injected pigs, by feeding plasma in the diet, it potentially reduces the viral load in those artificially infected pigs. We also collected the tissue samples and the lymph node from all the pigs, and the contact pig, and also the challenged pig as well. This slide shows the viral load in different tissues of the contact pig at the end of the study by different dietary type, plasma group and the control group. As you can see, the blue one represent the plasma group, and the yellow one represent the control group. A plasma group across the board in all the lymph nodes had a higher CT value, meaning they had a lower DNA copies, African swine fever virus DNA copies in all those tissues. So again, this is really suggesting by feeding a different diet, by feeding plasma, it actually significantly reduced the viral load of those contact pigs. So, in conclusion, we see a dietary strategy including inclusion of spray-dried porcine plasma really significantly reduced the African swine fever virus load of pigs in those tissue, in those lymph nodes. Also, it reduced the virus load in the blood and nasal swab and rectal swab as well. And also we see by feeding this spray-dried plasma, it can delay the infection on the onset of these clinical symptoms or in the contact pigs. Really, this study really scored our concept about the dietary role in helping pigs helping pigs not get infected or reduce the risk of infectivity. Of course, plasma it's an ingredient with a high biological efficiencies. With all the ingredient that has the high biological efficiencies, we all need to make sure it's safe as well. So at APC, this is the safety. It's really what we do, and there are a lot of steps we put in place to make sure the plasma safety. And here I'm listing a few of them. First, we're really collecting blood from healthy animals, and those animals has to go through federal inspections to go through to be processed, and those animals are fed to human consumptions to be able to be processed at the facility we're collecting our blood. And also at our manufacturing facility, we go through several steps really to make sure, really redundant step to make sure the safety of our product. Those steps include the UVC treatment, spray dry, and post-dry heating treatment. By applying those steps, we can absolutely make sure 100% inactivations of any potential microbe in plasma. In the last year, we have engaged University of Minnesota did a risk assessment about this multiple hurdle spray dry porcine plasma productions. And what the Minnesota group, the veterinarian group, has concluded is our manufacturing process would have sufficient inactivations under all calculated scenarios. So we have very efficient, sufficient, redundant procedure in place to make sure of the safety of our product. And this is according to a third-party assessment. So in conclusion, dietary plasma inclusion improve swine health, herd health, and also improve the resilience of disease challenge. And dietary inclusion of plasma reduces African swine fever virus load in blood and the virus of tissues of infected pig, and also help to delay the infections of those contact pigs. So also we believe dietary strategy are very important, especially those dietary strategy improve gut health and directly impacting immune system. Those strategies need to be considered for African swine fever virus control. And plasma protein manufactured using multiple hurdle technology are a safe and effective functional ingredient for pigs for helping their health. Producers in the African swine fever zone should really consider this technology for mitigating the African swine fever threat. With that, I would like to thank you for your attention. I appreciate your time this morning.